might be a day he wants to watch it. So there we go. All right. So main points we've covered so far. Friday's lecture won't upload, and I'll email the exam Monday morning. So it's due Tuesday at 11:59. And then the other thing we have to talk about is the group presentations. So I've heard from most of you, almost everyone. I won't tell you if you are among the ones who have not. But here's how it's looking right now. So we've got 4 people who want to present the virus evolution, which would be the paper we discuss next on the 13th of April is the day that it would actually be discussed, but then you'd have to be prepared on the 10th for questions from your classmates, like we've been doing the whole time. You can see that everybody wanted to do antibiotic resistance stuff, which is interesting to me because we haven't touched on it at all. Only 2 people want to do the paper on insecticide resistance and all that, but I need the groups to be about 5, so that means that a whole bunch of people who have antibiotic resistance as their first choice need to move to a different topic. And I want to be fair. Yeah, Benji?
Shit. I'll move to virus evolution if you need me to.
Yeah, that's great. Perfect. And then the other 4 or other 3 people need to decide if they would be willing to do the vector evolution stuff.
I'll do the vector evolution.
Yeah, I can switch to that too.
I'll switch too.
Okay, so I didn't expect you guys to be so willing.
They're all cool topics.
Yeah, I think that it's really true, and I feel like antibiotic resistance gets a lot of attention because you guys are familiar with it. Virus evolution is what we're doing right now. It's this weird thing about vector evolution you guys haven't done yet that makes it a little bit strange. So who said they would do vector evolution? Sydney, Alex, and Jarrett. Good. That means, here, wait, I'll edit it so you can actually see what we're doing now. Hold on. Oh my God. It won't let me escape. All right. It's so weird that Zoom messes with the controls of PowerPoint. It's like I hit escape and it advances slides, so it's very strange. And then it was Sydney, right?
Yes.
And it's moving you guys around my little panel too, so I'm looking in the wrong place. Alex, Jarrett, and then if you're not on the list now, you're going to do antibiotic resistance. So that would be Christy, Isabel. Who did I forget? Nicole's done, Isabel, Benji, Madison, Christy,
Lucy,
Kayla, and Lucy. There we go. Sometimes I look at you guys and I'm like, but you normally don't sit there. And it's not like the 2 tables. Like it's not asking you like the 2 tables in the classroom did. So it's a little bit of a brain melt for me. Okay, so that should do it. And so I'll save this, and when I upload it, you guys will know. The papers are all on Blackboard now. If you've looked at this previously, there were old presentations in PDF form and old handouts. And normally what we do is when we get these presentations ready, you guys put together a few slides. Normally it's just the figures from the paper unless there's something more you want to explain. And then you put a handout together like a reading guide that I've made, okay? To do that, of course, that means that we have to have separate meetings probably with me when you guys have read the paper and can talk about it. So Madison, Isaiah, Landon, Derek, and Benji, you should look at those papers sooner rather than later. This is the virus evolution one. The reason there's 2 is there's a response paper that basically argues against the conclusion of the first paper. And so it's an interesting response, and I think we're already starting to see people doing the same thing with the COVID-19 virus, and I think it's really interesting and fun. It's kind of fun to see the way you put together an argument. Okay, all right, so what we did on Monday, if we don't remember, is we looked at how phylogenies help us understand the evolutionary history of pathogens. And what we figured out from that paper is that phylogenetics can tell us the source of the pathogen, so what animal is it from? We can then also look at how a given pathogen is related to other pathogens that cause disease in humans. And then we can also look at pathogens that have this similar genomes, right? So with viruses especially, the components of the genome, which genes are actually present, is really important to whether or not it causes the same types of symptoms and disease. So that's what we got from the paper. And it also, I think one of the cool things about this is it allows us to sort of get at Christy's questions about what exactly is, how are we coming up with drugs, right? So the fact that we have the ability to take a genome from a virus and compare it to another genome in a virus means that we can then have some sort of similar topics or targets of medicines. So I think Christy asked a couple lectures ago about this rumor saying that there are antiviral drugs used to combat HIV in being targeted to look at COVID-19 or at least treat COVID-19. And so we had discussed that it's potentially what they call nucleotide analogs. So these are individual nucleotides that mimic the effect of a normal nucleotide. And so when we look at these two viruses, what you should see on this screen are two images. On the left is SARS-CoV, so the original SARS outbreak from 2003. And then on the right is HIV. So both of these are RNA viruses. The difference, there's actually a big difference between these two types of viruses. So the SARS viruses are single-stranded RNA viruses that can be translated as soon as they get into the cell, right? And so we talked about this previously where the RNA comes out of the virus package, ribosomes immediately translate it, they make a bunch of proteins that produces this RNA-dependent polymerase that makes new RNA that can be packaged and sent off into a new virus particle. Right, and then, so there's a whole bunch of copying the DNA, or sorry, the RNA, and then making the proteins from that RNA to make new virus particles. In contrast with HIV, HIV is an RNA virus that has two strands of RNA in it, two separate single strands, and also within the virus particle is an enzyme. So it's a protein already made. And it's called a retrovirus because that RNA is reverse transcribed back into DNA. So this is very different than just entering a cell and just making proteins from it. This has to be first converted back into DNA and then made into RNA to make the proteins for the virus. So what's surprising is that the first thought of a lot of people was to start treating COVID-19 with HIV antivirals because they're so fundamentally different, right? Kayla, is that a hand up or was that just a pencil up? Okay. All right, so when you think about these two viruses, really the part of this that that you're actually interested in comparing in terms of treatment is if you're going to use something that treats HIV, what actually is similar? And the only real thing that's similar is, in fact, the nucleotides, the types of nucleotides they use. But one thing you don't see very well in this, so on the SARS-CoV side, there's a side that says proteolysis or proteolysis. And then on the HIV side, it says cleavage and assembly of viral proteins. All right, so one is on the left side in kind of blue blobs, the other is just in black text all the way over to the right. Okay, so when we think about designing drugs, there are actually evolutionary principles that come into play here. One of the first things is that I'm showing you a diagram of replication of SARS-CoV, which is not the same virus as SARS-CoV-2. So one of the things we're inferring from evolutionary biology is that since they have the same genome, they're making very similar genes, and the genes and protein products they have are what we call homologous, right? They have the same things because they have a common ancestor. So they have the same gene elements, and they have the same proteins because they have a common ancestor. Therefore, when we design the drugs, we can design them based on homology of the two viruses. The other way we can think about evolution being informative is the idea of convergent evolution. So what we talked about in the paper on Monday was that it's possible that the similarities in protein structure of pangolin and bat viruses is actually convergent evolution, right? So in any case, what both of those things are doing, homology and convergent evolution, is finding similar solutions to enter the human cell. Okay? So that's how we can use evolution to inform making new drugs or treatments. But the other way that we make new drugs and treatments is purely mechanistic. And that is, we know they do the same things. Therefore, we're going to try the same medicines. And so what they're doing with the HIV and the SARS comparisons is saying both of these guys require proteases and both of these require nucleotides. So can we mess with the nucleotides and the proteases? Okay. So I found a few things. So there's kind of two ways we can do this. We can either treat the symptoms of the virus or symptoms of the disease caused by the virus, or we can actually prevent the virus from replicating. So when you use an antiviral med, what you're doing is you're preventing viral replication. And so what's shown here is actually an RNA virus, a hypothetical RNA virus, and the different steps it requires. This isn't specific. This is just showing you that there are all these potential ways we can target viral replication. So anytime you see a red dot with a line that's flat, it's supposed to be saying this particular drug would stop the step that it's next to. So if you look at the top left corner, you see it says none with a red circle, then the line, and then it's flat. And what it's saying is this step would prevent attachment of the virus to the cell. And you can walk around and look at these, and there's all these different steps. So you could prevent attachment, you could prevent fusion of the viral envelope, uncoating of the virus, you could prevent translation of the RNA, you could prevent replication of the RNA. And you can walk around the whole thing and see what the possible things are. Now, when we talk about attachment is going to be specific for every virus because whatever is binding to the surface is going to be dependent on the shape of the virus protein and the shape of the receptor it binds to. So SARS and SARS-CoV-2 might have a similar thing we could block. But in contrast, if you look at something like stopping translation, that could be something really general. It doesn't have to be as specific, okay? So that's what this diagram is supposed to be showing you. And what you can see is that in the places where we have drugs that exist, you have a list of drugs showing you which step they interfere with. Okay, so boceprevir, tenofovir, sinoprevir, etc. Those are all blocking some sort of protease of the virus. So the proteases, remember, are enzymes that break up large polyproteins. And if you remember in SARS-CoV-2, even in HIV, when they do the translation of the genome, when the virus's genomes get translated, they have to break up these big polyproteins into their individual function. So proteases are often a target. Another big target is down here with the RNA-dependent RNA polymerases. And what they're doing is stopping replication of the virus. Okay? And you should notice that normally when we name a antiviral, it ends in VIR, typically ACVir or EVIR or AVIR. So you can kind of hear it when somebody tells you that's what they're giving you. Okay? All right, so this is the general idea of people who design antivirals go with something like this. Which step am I going to prevent? And the reason that we find certain things that are good and certain things that are bad is that they aren't co-interacting with the host. So it's much easier to target an enzyme we don't really use that much or a protease we don't typically have, right? Because it doesn't cause harm to us. Okay, so here are the drugs currently being tested that I can find right now. The ones with the little asterisks next to them are all antiviral drugs. So the one used for HIV that got the most press was lopinavir in combination with ritonavir. Lopinavir is a protease inhibitor used for HIV, and the ritonavir is actually doesn't really have a target for the antiviral per se. What it's doing is it's helping lopinavir last longer in the cell. Okay, so unfortunately, like a week ago, the clinical trials for this with COVID-19 patients showed there's no advantage to giving lopinavir and ritonavir. So these are kind of like out the window. They're not going to be even tried. There's another HIV protease inhibitor in the trial called duranavir, and I can't find the research on that yet. So it's currently undergoing treatment. People are trying it and seeing if it's working, but we don't know about it yet. Another one that they're considering using, and the trials will start in April, so this month, is remdesivir. And this one is an A analog, a nucleotide A analog. And it doesn't immediately stop translation of the RNA, but it definitely prevents it from completely copying the viral genome. So it's not like when you add the A, it stops altogether. It causes it to terminate it causes the RNA-dependent RNA polymerase to terminate early, so it's not making as many copies. Okay? So the funny part about this is, right, so there's this hydroxychloroquine that supposedly is working based on very small trials. It is not an HIV med. It is an anti-malarial. Malaria is caused by a eukaryotic pathogen. And if you look at the review papers that are out there talking about what drugs are being used, they give you very specific details about what the drug is supposed to do. So for example, lopinavir is a protease inhibitor, specifically inhibiting the aspartate region of this particular these proteins. So when you can go through the article and see exactly what it's going to do, it's great because you know, all right, well, this makes sense, especially like remdesivir, you know, okay, well, this is going to be replacing all the A's and it seems pretty resistant to the virus responding great. But with hydroxychloroquine, basically what they've listed as the possible modes of action is everything on this figure. So it might prevent attachment, it might prevent fusion, it might stop translation, etc. So basically we have no idea if it's really going to work, and we don't even have a proposed way it could work. So it's in trial now. I think it's supposed to be done in a month, but we'll see how that goes.
So why are we using it then?
Why are we using it?
Or wanting to use it, I guess. Yeah.
So in a very small trial in France, a researcher gave it to, I think, 20 patients. And it was given alongside azithromycin, and several of the people who took it recovered.
What I read is that it was a little bit bigger than that. It was like 65 or something like that.
Yes, only 20 of the data points could be used of those 65.
Wow.
Okay.
So it's not a controlled trial. It was something they're doing now because some people seem to be responding to it. We just have no mechanism of action or proposed way it's working yet. Yeah, Benji.
So what made them say, okay, we have a viral disease, let's give anti-malarial drugs and antibiotics to it?
Well, I think part of it is treating, the antibiotic is partially treating post-infection pneumonia. That's essentially bacterial.
So like secondary diseases.
Yeah, and there's some evidence that hydroxychloroquine helped with SARS infections and MERS infections.
Oh, gotcha. Okay.
So it's not like, I don't know what the first inspiration to try to use this was in these types of infections, but it's possible. The reason it got accelerated FDA approval is because Trump asked them to accelerate approval for it.
I have a question.
Yes.
Sorry, I can't figure out how to raise my hand on here without my video being on. So hydrochloroquine has also been used as a treatment for lupus. So could it be that they thought that maybe it could help with like immune boosting?
So one of the other things about treating this is that what seems to be killing the patients is when the immune system goes out of control into a cytokine storm. And so it could also be that what hydroxychloroquine is doing is knocking back the host immune system. We just don't know. And it is approved. They're using it for, what are they calling it? Compassionate use. It's approved to use that. So if someone's in really bad shape, they can give hydroxychloroquine, and the drug companies are in fact synthesizing more of it because I guess that a lot of people want to try to use it when they're in really bad shape. But there's currently no good trial. Well, it's ongoing. The trial's set up now. It's ongoing, and they are going to test it and see if it's improving people. So that's something. It's possible that it does have antiviral properties, and we just don't know it yet. They're also testing chloroquine, so just remove the hydroxy from it. Chloroquine's a little bit more dangerous. It has more side effects. It can cause permanent brain changes. It certainly causes hallucinations so when you do a lot of work in the tropics, they often give you this prophylactically if you're going to be exposed to malarial and transmitting vectors. And most certainly the common reports are things like hallucinations, night terrors, and really horrific nightmares. So it does have some effects on the brain, the chloroquine, not the hydroxychloroquine. So, the side effects are, I think, the question is whether or not the side effects are going to be worse than just recovering from COVID-19. So that's going to be the question. On a more positive note, there's something that's actually been in the works for a while for SARS. So when this virus enters into your cells, it binds to this receptor called the ACE2 receptor. And this is a receptor, if you remember, found on a lot of different epithelial cells. And clearly this is being done by Germans because everything's written in German and in English here. And I kind of hope that if they publish it, they use something like these words because they sound more fun than attachment, right? And Heftung. I think that's way more fun to say. But what they're trying to do is, so when the virus binds to ACE2, we have a protease on our cell membranes called TMPRSS2, transmembrane protease serine 2, and it cleaves the spike protein. And when that cleavage happens, that allows the virus to enter the cell. And so what the drug, this one's Camostat mesylate, that's down at the bottom right of this. This one actually targets TMPRSS2, so that's on our cells, and blocks it from activating the virus and allowing it to enter. And so this is supposed to be completed by May 1st, if this is an effective drug. So that's kind of cool. And this one seems like a little bit more promising because it's had in vitro trials. And I mean, I'm not saying that the other ones have not. So the reason that the HIV drugs were used is because it showed that in cell culture and in mice, it worked. It prevented infection, but it didn't work in human clinical trials. So we have hope, but we don't have any answers yet. This is another one that this is like a purely mechanistic approach, but it's also evolutionary in the sense that was designed for SARS-CoV, and since that spike protein, the mechanism of entry is still the same, we can actually target something that is meaningful. So it's sort of positive, it's sort of scary in any way, because I think the amount of time it takes to approve a drug is pretty intense. So, oh, I think this is supposed to say May 2020, not May 2021. Sorry, guys. That looks even more depressing. Okay, so Christy, I hope that answers your questions about what's going on with the drug delivery.
Yes.
Good. But I also want you guys to remember that, like, the reason I'm doing this is not just to help you guys understand all the stuff that's out there, but it's also because a lot of this is rooted in understanding evolutionary mechanisms of how viruses evolve, right? And so we have some targets of hope because of the way we think about questions in evolution. Okay, so what we need to do now is talk a little bit about, apparently everything all over again, is about phylogenetics. And I realized that when we started talking about phylogeny, it was in originally in the human papers, and then we started talking about it again when we were doing the blood type papers and all of that. And I want to make sure everybody is clear on a few things, because phylogeny I assume people get, and then I'm not sure. So on the side, on this slide, what I've done is I've just taken obviously frog phylogeny, because it's pretty more than anything. It's not anything other than it makes a nice phylogeny. And the things on the right side are things that you should be able to do with a phylogeny. So you should be able to identify what a clade is. You should be able to explain why the branches are long or short and what it means. Last time we talked about the numbers on branches. I think we did that on both Friday and in the discussion of the virus on Monday. And then if I were to ask you, I've labeled a few of the clades with letters, or the base of the clades as letters. Which of them evolved, which of them happened first? And I really want to know if you guys know this. You can answer in text if you want to, like you can text on here and answer. That's easier. Oops. You got to see it to be able to answer it, don't you? Does anyone?
I feel like it would be A, but
Wouldn't it be
I
A?
don't know.
So well, let's talk about this. Where's time on this way? Which way does time go?
To the right.
To the right. So if I was going to annotate this and draw, I would do this, right? And handy, it actually made an arrow. I don't even know how I did that. Cool. So time, oh boy. So time is shown by the arrow, and this would be older. And then this would be more recent, right? So that means A is the oldest event, or the one that happened first. Then you get a change. Then it would go B, then D, and C probably about the same time, right? So there's some assumptions about that. So let's go back to the second question on this list. Why are the branches different in length? What do they represent? Benji, I can't hear you. I think you're on. Oh, I can unmute you. I can't unmute you. You have to do it yourself.
Isn't it like how different they are, like how many nucleotides are different?
Yeah, so I didn't tell you this was DNA, but it is a DNA phylogeny. So it would be how many nucleotide substitutions there are, right? So one thing to remember is that on these branches at the back, right? So the branch that goes from, I'm gonna make sure I label this right. So from this check mark to this check mark, that means that everybody after those branches has the same substitutions. So it means that, like, if there's an A, and a T, that everybody would have that together, right? And then that means that, trying to figure out how to do this, you guys, it's like too many things happening at once. Okay, so that means that a branch shown at my little X here, that branch would have all unique changes there. Anything there would be unique to Discoglossus galgani, or galganoi. right? So we can actually make some inferences about that, all right? So what we're saying then is that this changed to A at whatever position that is, happened a long time ago, and then these up here are probably more recent. Does that make sense? Okay, all right. So that's some basic things. So if you're still struggling with those concepts of, I'm going to show you what a monophyletic group is in just a second. I'm going to turn off all this annotation. Okay. And so, yeah, just to make sure A happened first, yes, the numbers on the branches reveal how much support there is for those branches. And when you see two different ones, it usually means they estimated support in multiple ways. Okay, so in this particular case, we know that the value in the parentheses, as people who study phylogenetics, we know that these are, the way that these are done always gives them more support than it probably should have. And that's an artifact of the method. Okay, so just to clarify some things about monophyletic groups, I've got to clear all of the annotations here. Oops. All right. So if you've forgotten what a monophyletic group is, it's the ancestor and all its descendants. So the correctly labeled monophyletic group in this diagram is in blue, and it includes H, I, J, and all the ancestors within the blue shaded box. You can also describe things. People who talk about phylogenies a lot like to name things like, when you don't include all of the descendants, it's a paraphyletic group. So the ancestor is at this base of the pink box, but it doesn't include all its descendants, and we call it a paraphyletic group. If you don't include the most recent common ancestor of a group, we call it polyphyletic. So the most recent common ancestor of E, F, and G is all the way back at the base of the tree, so we would call that polyphyletic. For me, it's more important that you recognize what is a monophyletic group and what is not. But if you ever see these two terms, paraphyletic or polyphyletic, you know it has a specific meaning, okay? All right, what else is written on this slide? Oh my God, look at that, I'm drawing lines everywhere. Okay. All right. So one of the things that's come up as problematic in the past of understanding phylogenies is that when we talk about common ancestors, you can see that every one of the letters on this phylogeny has a common ancestor. It's just all the way at the back, the base of the tree, right? Oh my God. So back here is the common ancestor of A through J. But we often then talk about the most recent common ancestor. So if I were to talk about the most recent common ancestor of A and C, that would occur where I'm circling, okay? That's the most recent common ancestor of A and C. It's not that the one at the base of the tree here isn't common to A and C, it's just that it's not the most recent one, okay? Everybody good with that? Okay, so we use this terminology a lot, and we use it, maybe we name too many things, but that's just to give you some basic definitions in case you like come back to it and then worry that you're not sure of it. So, okay. Then we talked about on Friday, this idea difference between a substitution rate and the mutation rate. And we talked about mutation rate as what was happening within an individual. And then we talked about substitution rate being how it differs as it crosses to another individual host. There's another way to look at substitution rate. And people love this kind of drawing shown here with all the colorful circles. And so when you look at the colorful circles, what you see is, in the case of viruses, it would be an individual with a virus, so it would be a host. But it also could represent evolution within the host, if you wanted it to. But we'll just do it simply here and talk about it instead as humans, right? So if each circle at the bottom of the diagram represents an individual host or an individual human, you can see that everybody has the same genotype because they have the same color. Okay, does that make sense, everybody? So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 humans down at the bottom. And then the connections represent transmission to a new individual. So this would be offspring of humans. So if I look at this one, they have 2 offspring here and here, and they have the same genotype. Does that make sense to everybody? Okay. Then when you change color, what you get is a new mutation. So this individual has 2 offspring, and one is different than the other. Right? Everybody good with that? So you can follow this all the way down. If they don't have any connections like this one here, they go extinct, right? So what ends up happening is this teal or grayish blue lineage goes extinct, right? And by the end, if you follow this through time, what you end up with is seeing the variants of species A, the variants in species B. There are 2 yellows there. I don't know how well it's showing up on your computer. And there is 2 different variants of C. Okay, does that make sense? There are no nods happening.
Is the split right here like isolation or something, like geographical isolation? Or
It
something?
could be. It could be. If you're thinking about it in a simple speciation model, yeah, it could be. And what you notice there then is that species A and B initially aren't different from one another, right? When they first are geographically isolated, they're not different than one another. Okay, so there are a lot of things we can talk about, but one of the things we can talk about is that all of the colors at the tips of the tree have a common ancestor back where I'm going to write the letter A here. And we refer to that point as the coalescent. And it's an annoying term, but it's called the coalescent, where you can take all the variation in these 3 species and trace it back to one mutation, that blue mutation. So for some reason, all of you guys are on mute today. So if you have something you want to say, you have to unmute yourself. So I see mouths moving and no sound, and that's okay if that's intentional. But if you think I'm hearing you, I'm not. Okay. All right. So when we look at the difference between a mutation rate and a substitution rate, one of the things that we talked about was mutations happening within a generation, and substitution rate is happening across generations or across transmission events. But they're also influenced by selection, the substitution rate, and by changes in frequency. So when we talk about mutation rate and substitution rate, we have to include frequency. Okay, so one way to look at this is, if you look at the pie charts down at the bottom, you can talk about types of mutations you get and their effect on fitness. So red would be highly bad for you, pink is slightly bad, gray is neutral, green is slightly beneficial, dark green is really beneficial. And so there's a relative frequency based on the pie size of how often those occur in mutation rates. But in substitution rate, it's a little different, because it's not just the size of the slice of the pie that matters. It's about how quickly they occur. Right? So really beneficial mutations are going to happen at the same frequency, but they're going to happen more quickly, because if it's beneficial, that means they get passed on more frequently. Does that make sense to everybody? And you see how that pie chart on the substitution rate side actually shows you that you get them at the same frequency, but they happen more quickly because of the fitness effect of the mutation when it's beneficial. Does that make sense? Whereas you don't see the lethal ones because once they happen, the lineage just disappears. Yes? Either all of you have frozen. Wait, now you're moving again. It's like I can see frozen movements in the slides. It's awesome. Okay, so when it comes to talking about the paper that group 1 is going to present, substitution versus mutation rate is one of the things that authors are going to argue. So we can talk about this when we meet and then present it later. But these are some things that we should consider. So when you think about the fact that COVID-19 all has the exact same spike protein and that hasn't changed, that's because that mutation happened and then it fixed in the population of COVID-19 or SARS-CoV-2. Okay? I'm a little worried that the transmission is bad today. Like our internets are slowing down. What's happening on my text? Okay, all right. Okay, so I thought I changed this. Hang on, let me just get out of here for 1 second. All right, so that told you when we talked, what time is it? Haha, I knew it. So I told you that on Monday, we would start talking about Benji's question about how do we get these new forms of viruses and how do we get novel viruses. And so one of the things we need to talk about is this idea of coevolution and what we see when species co-evolve. And I'm going to define coevolution and then we're going to stop for the day because it's about that time. But when we talk about coevolution, we talk about it's an interaction between two species. And when one species changes, the other changes in response. Okay, so what this diagram is just funny. So it's, here's this host, this human host who's running on a treadmill. And every step he takes is supposed to indicate a change that gets him away from his pathogen. And then here, this surprisingly green pathogen would then develop a mutation in response to evade whatever the host has evolved. Okay? Does that make sense, everybody? So there's a specific thing here happening. It's not just that one species evolves. It's that one species evolves to evade this pathogen, and then the pathogen develops a mutation that allows it to get over whatever the host has evolved. And so we have that same idea of the Red Queen hypothesis that we talked about, where there's this constant chase where every time the human evolves a new mutation to avoid a pathogen, the pathogen has a new selective pressure. Now, if it can get away from the host's immune system, it has a new response, okay? Does that make sense, everybody? Cool, all right, I wanna stop it here today because I don't wanna get into all the rest of this now. I think everyone's message, I know it does, it sounds like I'm really drunk, Jaquez, it really does. It's really funny, but I'm not, I promise. It's just water. It's not, it's just water. Okay, so hopefully it recorded okay. And I will get this up and uploaded. If the group 1 wants to schedule a meeting, send me an email. You guys know who you are, I think, just in case you don't. There we are. You guys should actually talk with each other and schedule some times to read the paper, do the paper, then talk with me to get when we're going to meet. Okay? Cool? Okay.
Should we just like make another group me for like our individual groups, or how do we want to do that?
If you want to, whatever works best for you guys. You guys know how you communicate with each other best. So.
Yeah.
And you can add me whenever you want to, or however it gets done. And
We're
then
all working together on this.
yeah, so each person listed under the topic is working together on their paper.
Oh, okay.
Does that make sense?
Yeah.
And that paper is listed with these titles on Blackboard under the research papers. I don't want anybody to have to present a paper on their own in this class. It's too hard. Okay,
Okay.
All right, guys. Shoot me emails or group me, whatever, if you have any questions.
Okay.
All right. Talk to you later.
Bye.
Bye-bye.